I usually like to spend a little time talking about the evolution and how you can make predictions about evolution and mutations from sequences. So this is protein sequence. Each letter here represents a single amino acid in a protein. And this is protein sequence from 4 different organisms: human, zebrafish, both vertebrates, and then sea urchins and brown algae. So I think you guys would agree that these are pretty diverse um, organisms. They're all eukaryotes, but otherwise pretty diverse organisms. And yet, if you look at the amino acid sequences between these 4 diverse organisms, you can see that this protein is very similar in these 4 diverse organisms. This happens to be a protein that generates a motor. Remember that uh, motor that drags the chromosomes along the mitotic spindle during mitosis? So that's actually what this protein is. And what this sequence shows us is that there are regions of the sequence that are conserved between these very diverse organisms. And this means that over evolutionary history, first of all, the fact that all 4 of these diverse organisms have this protein tells you that this gene was found in their common ancestor. So the common ancestor of these 4 organisms was a very early eukaryote. We've got animals and plant cells here. So the very earliest eukaryotes had this protein, had this gene, and could make this protein. And the fact that the protein has been conserved over probably about 2 billion years of evolution tells you this protein must be quite essential for the survival of eukaryotic cells. On the other hand, there are regions of the protein that have not been conserved. So the asterisks on the bottom indicate the parts of the protein that have not changed over about 2 billion years of evolution. And the spaces represent the regions that have changed. So some parts of this protein are absolutely essential for the protein to do its job and for the cells to survive, and other parts of the protein are less essential. So when we look at the DNA sequence, we're going to see mutations in those regions between these different organisms, because those regions of the protein are probably not as important. But the DNA sequence for the sequences that with the asterisks are going to be a lot more similar. Now, this doesn't mean mutations can't occur in those DNA sequences. Mutations happen everywhere. And so mutation is not, these regions are not protected from mutation. But mutations that occur in those sequences that affect the performance of this protein, that change the amino acids there and thus affect how effective the protein is, are eliminated by evolution. And so over evolutionary time, mutations that occur in those regions get eliminated. Now, there's one type of mutation that you would expect to find in these areas that wouldn't be eliminated by natural selection, and that's silent mutations, because evolution can't detect silent mutations. Those are those mutations in the 3rd letter of a 3-letter word. So the codons that specify E, R, M, V, N, Q, N, for instance. Those 3-letter words for each of those amino acids, many times you might see a mutation that's in the 3rd letter of those 3-letter words. So if we looked at the DNA sequence between these, we would see mutations have occurred in that region, but the mutations have all been in the 3rd letter. Mutations that occurred in other parts of that DNA sequence have been eliminated by natural selection. So you can really use DNA sequence and protein sequence as a yardstick of evolution and see where evolution has occurred, and also see how natural selection has eliminated some mutations because they're so disadvantageous. So we could predict then that the amino acids that have asterisks associated with them in this image are really important for the function of this protein, whereas the ones that haven't been conserved are probably less important.